So we have studied a lot of environmental factors which can affect the viruses, which can affect in the better way or which can harmful the viruses. Just like we have studied the temperature, we have studied the effect of the pH, we have studied the effect of different chemicals on the viruses. We have studied that the viruses can favorable temperature for the viruses is normal temperature or the room temperature, and similarly the extreme temperatures are not favorable for the viruses. So if we are going to research on the viruses, if we need to investigate about the viruses, we need to inactivate the viruses, we can use those environmental factors in our own way. Similarly, as far as the pH is concerned, the 7 is the neutral pH for the viruses, and most of the viruses will be favorable in this pH. Other pH, extreme pH, low pH, and the high pH will not be good for the viruses, and viruses will be inactivated in the higher pH. However, they can again be gathered in the normal pH levels. So now we will see the effect of the radiations on the viruses. In other words, we can say that how to kill the viruses with the help of the radiations. Radiation effect. First of all, we see that what is the radiation? The emission of the energy as electromagnetic waves are moving as a subatomic particles, especially the high energy particles which can cause the ionization will be called as the radiation. Ionizing, what is the meaning of the ionizing or the ionizing radiations? Lot of reproductive activity and the faster inactivation in the crude suspension will be called as ionizing radiations. Which can cause the viruses, how the radiations can affect the viruses? There are three different ways they can affect in the change in the structure. Of course, the viruses are made up of nucleic acid or simple structures. Also, they are the protein coat, so they can change its structure. They can change it. The radiation can change its structure. Changes in the genome structure can also be done with the help of the radiations. The nucleic acids made up of the chemicals, so therefore it can also change by the radiations. There are some little simple proteins are present inside the viruses, so changes in the proteins can also be done with the help of the radiations. Here is one of the examples of the radiations which are going to a virus. We have taken the example of the oncolytic virus. The oncolytic viruses are those kind of the viruses which can cause the cancer. So oncolytic virus is there. It is in viral receptors upgradation and the increased viral uptake. First of all, there will be the four kind of actions will be taken when the radiation is inside the oncolytic virus. Then the increased viral gene expression, and similarly specific transgene expression or the upregulation of some specific under the control of radio-inducible promoters will be done. Here you can see in this diagram. This we have taken the example of the oncolytic virus, and we are put the radiations on this virus, and we see this can show its effect by the four different ways: 1, 2, 3, and 4. And the number we have already discussed the three of those, and fourth one is the increased viral replication. So the tumor cell will be increased in the viral replication, and which will be very beneficial for the tumor cell. There are different kinds of the radiations which we need to study. First of all is the UV radiation, the ultraviolet rays. These are the exponentials, and survival particles show no reproductive activity after the ultraviolet rays has been demonstrated. And extensive changes in the genome sequence will be occurring with the help if apply the UV light to the viruses. There will be extensive changes in the genome sequence, and sometimes it will also inhibit the replication of the viruses. Here you can see we have 254.7 nanometer UV light, and this will be directly acting on the genome sequence. It is the double-stranded virus, DNA double-strand virus, and here you can see that it will directly happen on the nucleic acids and formation of double bond, and UV inhibits its replication directly. Then what other effects the UV, how can it affect the other ways? UV rays can damage the DNA of living organisms by creating the nucleic acid dimers. The nucleic acid dimers are very important, and they are the fake sequences, you can say, or the nucleic acid sequences are present, but these sequences are nothing to do with the normal sequence of that specific virus. The damages are not important due to low penetration. The UV lights is low penetration in the living tissues. However, they are inactivate the virus since the virus particles are small, so the UV lights can capable of inactivating the viruses. And they can also reach genetic material of the virus and can stop its replication. There are fake dimers are produced, and once the dimers are produced, the virus particles are not able to replicate further. UV light with the help of the riboflavin can be effective in reducing the pathogens in blood transfusion products. So it is very important from the medical point of view for the medical application of the UV lights. These riboflavin and UV damages the nucleic acid in the viruses, bacteria, parasite, and donor white blood cells, rendering them to unable to replicate and cause the disease. So UV light along with the riboflavin is very important from the potential of the medically point of view, that they can be used to stop it as in the donor white blood cells and rendering them replicate and cause the disease. One more example of the radiation effect of the viruses. Radiation and or the chemotherapy, the use of the chemicals we have already studied. It can of course damage the DNA damage. How many ways it can damage? Just like here, upregulation of the transcription resulting in the increased of the protein expression. Similarly, complexes with other regulators are the translations, and then the mediates the demospheric of the EF2A. So here is the diagram. This is self-explanatory, and you can see the radiation mediated enhance the viral replication here, and there are 5 different ways which are explained here.